All right, guys. Yeah, we are all there, right? We are all here in the meeting, I can see. Okay, shall we start? So the session for today, remember last time we did the prokaryotes and other stuffs, but today we'll do eukaryotes. All right. So let's start. Okay. So one thing that we need to know about eukaryotes, we talked about the the examples, everything that. Okay. So these guys, we said they are multicellular organisms. All right. They can be single or multicellular. There is one guy under them who is trying to fall the other side of prokaryotes, but not really that. The guy is called protist. So we'll call these guys to say, so we are talking about eukaryotes, by the way. Okay. So we are talking about eukaryotes. Okay, let me just light eukaryote. Okay, just like that. Okay, so we are talking about eukaryotes in particular. So since we are talking about eukaryotes, these guys, we say they can be single or what, multicellular organisms. So these can be single, right? Single or multicellular. I'm sure that is not important to light. So they can be single or multicellular organisms. So they can one of the basic characters or features that we can see with them, they will see some cells there of which we didn't see in what, in prokaryotes. So even some cell membrane, all right, cell membrane, we saw them. Okay, they were there also in prokaryotes. All right. So the other stuff that we can see or observe under this guy, we'll find also nucleus. So they are just same as prokaryotes, some things, but other things will differ. All right. They contain what we call endomembrane bound organelles. We'll see those organelles which has those features. All right. So these guys, they can go two types of division, and that is what we call mitosis. All right. And mitosis and also meiosis. All right. Okay. Okay, so those are the things that or features that we can see in these guys. Okay, so we gave some examples of which we said some of the examples we can have what we call all right, we can have what we call such as animals, they fall under this. As human beings, we have eukaryotes type of cells, and also fungi is one of them. So we said plants, all right, so fungi and also animals. All right, so these things have what, have this type of what cells, they are eukaryote cells. Okay, so we can have the basic structure of these guys. Okay, so the basic structure that we can give is let me light something here. Okay, so there inside, let me say I have a round thing there, then I have, let's say, like that. Okay, so then I have again something there, then I put some liquid inside there. Okay, then to this I'll attach something like this. Okay, okay, so I'll attach something like these are what we call ribosomes. All right, so these dot we can see they are called, by the way, labeling, you don't label, you just label a straight line, like not the way I've done it. So these I'll call them what, ribosomes. All right, so I'll say why they are ribosomes. So when we talk about ribosomes now, these ribosomes, they have some features. Okay, I'll come back to that. This is now an envelope of a nucleus, so I'll call it envelope nucleus. All right, envelope nucleus, envelope nuclear. Let me say envelope what, or just envelope pause. No, let me just say nuclear envelope. Okay, envelope nuclear. Okay, so that is it. Then the other thing that we can see here is I can put something here of which it will be just floating. Okay, this guy, I'll name it as what, Golgi bodies or Golgi apparatus, or just dicto or dictosomes, or like they are just dictosomes like that. So I can call them what, Golgi apparatus. Okay, so what I'll do is once I name the function, then I'll be what, I'll be able to explain. Or let me just explain these ones which are just on what, things that I still remember. Okay, so ribosomes will be there as well. Then inside there, the pores, you can see this is what we call now nuclear pore. All right. So it's a pore. Okay, so it's called nuclear pore. Then inside there's a liquid like those to my white thing, we call it nucleolus. All right, I mean it's not really nucleus. All right, so I'll say this is this gamma thing which we can see there is what we call now a nucleolus. All right. Okay, my handwriting is bad. So there, this is now a cell what, a cell membrane. Okay, let me just get to explain some part as we are what, as we are talking about them. So since we are saying the whole part inside this part that we can see there, the whole combination of this is what we call now what, a nucleus. All right, a nucleus that we have been seeing in different books since our what, since our grade 12. So these Golgi, I said these are Golgi apparatus. All right, so you can call them Golgi bodies or just dictomas. All right, so we'll call them so we'll call them dictosome like that. Okay, dictosomes. All right, so that's just another name. So these guys, they'll be there to collect, modify and transport food. All right, so they will be there to modify, collect and transport food. These guys, that's what they'll be doing. So they occur in what's these granules. So they can be found also in loads or just filament forms. All right, so I said they what, they modify. So they will be there to collect, okay, modify. So that's about Golgi apparatus. They operate apparatus. We know apparatus, they are just used for something, like let's say apparatus in the library, in the labs. Okay, so they will be also there to transport some things. All right, or some materials to say. Okay, so these guys, they will be there to transport these guys or these materials or made materials. All right, so that is it. Then what else can we do? Let's talk about cell membrane now. This cell membrane, it is actually formed like it has to my head there. They are fat like in nature. All right, that's why your skin sometimes, the moment you put what, it will dry very fast. All right, so that's one way to prove that the fats exist in the what, in the fats exist in the what, in the cell membrane. Okay, so this cell membrane, we can see it looks like this. This is a head. All right, then this is a tail. All right, okay, so since this is a head and this is a tail, this guy here, it is made up of two layers. All right, so it looks like this. It is made up of two layers. I'll put another things there. So it is made up of two layers. Okay, like that. So the head and the tail there. So this is the tail. So it is made up of two layers. So this two layer is collectively known as what? Phospholipid. Okay, so that is what? Phospholipids, like that. Okay, so it is made up of this layer. This layer, it is actually made up of what? It is made up of fat acid. Okay, fat acid and what? And lipids. That's the meaning of phospholipid. So lipids and what? Fat acid. So it is made up of that. So this head is what? Hydrophobic. Okay, so by hydrophobic, phobic simply means fear, so it fears water. Hydro is water, so hydrophobic. So there are people here who fears water. I know they don't like bathing. They can stay for 3 days without bathing even if water is there. So this tail there is what we call now hydrophilic. That's why if you can put even cooking oil in water, you'll be able to see some fats on top. Those are what we call now hydrophobic. All right, so hydrophilic are the one which goes down. All right, and those are the tails. Okay, so that's about them. So we can have different, I'm sure we know this structure. This will be a smooth endoplasmic reticulum. All right, smooth endoplasmic reticulum. Then this one which is there, it will be what? Rough endoplasmic reticulum, in that the ribosomes are there found on this guy to trying to what? To synthesize proteins. So ribosomes will be there to what? To synthesize proteins. So let me not even make, I'll be drawing them where possible. I'll be drawing them. All right, I'll be drawing them where possible. I think I should just put, we know the basic everything. The mitochondria, they won't bring in an exam, they won't bring, especially at CBU, they can't bring a diagram unless otherwise they have never asked this, bring the what's this the structure. So it's important that we concentrate on the what? On the on the parts. So we were talking about cell membrane. One of them, we said we have talked about the structure and also the structure, the way it is made up of. The other thing about cell membrane, now the function will be there for communication when cells are just neighbor cells. So the cell membranes will be there to communicate that, no, can we heal? Let's say we have an injury. Okay, so let's say someone is clotting, then the cells will be there to communicate within two. So we'll say cell membrane will be used for communication. All right, so this communication is for the skin. All right, so communication. All right, okay, so communication, so it will be used for communication because, all right, it is what? It can interact with other neighboring cells. Okay, that's about it. Then the other thing is it contains some proteins for transportation. When two cells are here, it will have some proteins there that can be transmitted to the other side of the cell. So the cells do help like that to help what? In what? Retaining energy or forming energy. So it will be used, so it will contain, all right, contain proteins. All right, proteins for transportation. So it has proteins with it, then it will transport to the neighboring cells. So for transport, for transportation. All right, okay, so that is it about these guys, about cell membrane. I'm sure the other thing that we can talk of cell membrane, we said it will carry that proteins and also active transport, because active transport is when minerals or ions are within it, then it can be transported into that form, and that's what we call active transport. So transportation of mineral ions. Okay, that's about cell membrane, guys. That's about it. So the other part that we can talk of is the nucleus itself. Okay, so we'll talk about now the nucleus. All right, so nucleus, we saw the structure of nucleus. It looks like this. All right, then it looks like that. So this is the nucleus. Okay, in case you didn't know, then as I'll be explaining, you'll be able to know some things. Okay, so within it there are what we call now nuclear pores. All right, so we can't just see them, but they are pores like whereby they can what? they can let things pass through it. Okay, so inside there is what we call now a what, nucleolus, like we said. Okay, nucleolus. Then outside there, this layer is what we call now what, that's where we find the chromosomes. All right. Okay, so since those are chromosomes, then what about these? These are what we call now nuclear pore. All right, nuclear pores. Okay, so since these are nuclear pores, we'll do what, we'll have, what else can we have? So I've talked about chromosomes, then being what, and just like that, envelope. So this is now the what, the nuclear envelope. All right, the outside thing is the nuclear what, envelope. All right, so since this is the nuclear envelope, now what are the function of this guy? So this guy will be there to do what, will be there to help us in what's this, controlling all the activities. More like it's the head, the brain of the cell. So to control all the activity, so controlling all the activity. Okay, so controlling all the activity. The other function that we can give it, it will store what we need for the what, for the difference of other our replicates, or just our offsprings. So it will store what we call genetic material. So it stores genetic materials. All right, so that's about the nucleus. It's a very important organelle in the what, in the cells. Without this, will not be, what's this, we can't have same things, I mean having them on the same land, everything like that. So that's about this one. Okay, 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 so that's about what, the, okay, that's about the nucleus. Okay, so we can lab them. So we have talked about the nucleus. Let's talk about something else. So we can also talk about endoplasmic reticulum. All right, so this endoplasmic reticulum, so endoplasmic, all right, is reticulum. All right, so this endoplasmic reticulum, they will actually provide a mechanical support for the cytoplasm. They will be found within cytoplasm. So once they are found within cytoplasm there, all right, this cytoplasm, they will be there. That's where we'll find them. They will be just floating like that. So they provide the mechanical support. So one function of these guys, they will be there to provide support, all right, or mechanical support. All right, about endoplasmic reticulum in general, they what, but we divide them, but they support what, mechanical support. Okay, mechanically. All right, so they support mechanically, and they will be there to support they'll be there to support the cells, or just to make it or maintain the structure of the cell. So that's about the that's about the endoplasmic reticulum in general. That's about in general. Okay, beside that, they will be also there to transport materials, all right, from one part of the cell to another. So they will be there rotating, since they are just floating and they can provide mechanical support. It's more like they are strong. Okay, they are strong on their own, just like that. Okay, okay, just like that, they will be like that. So some things will be with the ribosomes, then the other part won't have ribosomes. So they will be there to support mechanical support, or support mechanically. Then they will be there to transport what, to transport materials. All right, so these guys, they are just there in the cytoplasm. All right, so they'll be there to transport materials. Okay, so they can be divided into two, that is what, and smooth endoplasmic reticulum, and also smooth endoplasmic reticulum, and also laugh endoplasmic reticulum. All right, so what is this? So we'll start with the laugh one. So the laugh ones are the one which have what? Liposomes. You know, something which is laugh, it is distracted with, let's say, with this some kind of surf, sand, I mean to say. So these laugh endoplasmic reticulum, they have what? Liposomes with them. So in actual sense, okay, so down there, then that's the what? The nucleus there. So down there, that's where what? We find liposomes. So these liposomes, then this one, the down part, is what we call now the what? The laugh. All right, so then they will find another part just somewhere, all right, on top there. All right, this part there is what we call now smooth. Okay, so that is it. Okay, then this is the nucleus. Okay, so since these guys, they what? They are laugh, they have ribosomes with them. Okay, so since these ribosomes, we know the ribosome to say they will be there what? To facilitate or synthesize proteins. So it has ribosomes. All right, so it has ribosomes with them. Then the other thing, they will be what? Synthesize these proteins. So ribosomes will be there to what? To synthesize proteins. All right, that's the function of ribosomes, so to synthesize proteins. Okay, I'm sure that is clear. Then what about just for smooth? Smooth endoplasmic reticulum. Okay, let me just like that. So smooth endoplasmic reticulum, these ones will be there to do what? They don't have liposomes like you saw in the previous structure. So the function, they will be there them to facilitate what? Lipids, not proteins. Proteins are laugh in nature, so lipids are smooth. So these ones will be synthesized, lipid synthesis. All right, so they'll be there to make what? Lipids. The other thing that we can give, they will be just there to facilitate what? Facilitate other what? Biological reactions. Facilitate other reactions. All right, so that's about this smoothie. You know, the other thing that they can undergo is they'll be there to help us in detoxing substances. You know, detoxification is just a toxic substance. It's more like antitoxic. Okay, so they'll be there for what? Detoxing. All right, or a substance that are harmful to make them less harmful. All right, that's the meaning of detoxification. All right, so that's about these guys. Since now we said they have, the laugh ones have ribosomes with them, then what are the function of those ribosomes with them? I said one function of ribosomes is to do what? In fact, ribosomes are the one, are the smallest organelles in the cell. All right, so ribosomes will be there to do what? So we are talking about ribosomes now. Okay, so ribosomes are the smallest organelle in the cell, so they are the smallest. All right, so since they are the smallest, what we are going to do is those ones, these ones, they are found on the laugh endoplasmic reticulum. All right, what are the duty, what are the law there? What is the law there? The law is what? To synthesize proteins, like we discussed on the what? Laugh endoplasmic reticulum. So they are attached in the laugh endoplasmic reticulum, or they can be just they can be flowing, apart from being attached on the laugh endoplasmic reticulum. They can be just flowing. They are small, like they are dot like this in the what? In the cell. All right, so they can be flowing there, you know, just like we know other people there here at CBU. They will be flowing, like gallivanting around. All right, So these ribosomes, will find them in both eukaryotes and what, so eukaryotes and also prokaryotes. So in eukaryotes, they are about what we find the type of ribosomes which are what, 80Ss. So they are just called 80Ss. I don't know the meaning of S, but they are called 80Ss. Then in prokaryotes, so this P means prokaryote, so they are found, they are about what, a 70Ss. Okay, so that's the name of what, ribosomes which are found there. Okay, so function, they'll be there to synthesize proteins. Like I said, I'm repeating the same thing. Then okay, so apart from this, these guys, they'll be there also to attach what we call RNAs, especially RNA molecule. So RNA, ribonucleic acid. All right, so they'll be there to what, to attach these guys. All right, so once they attach this, then combination or genetic formation will be very effective. So when these guys are there, I said they are the smallest guy in the cells. All right, and they have RNA and proteins with them, because that's what they want. They attach RNA and proteins, so they attach RNA and proteins. So we can also go and talk about this guy called what, the one I just explained on the diagram, which is Golgi, all right, apparatus. So these Golgi apparatus, we said they are apparatus. They can also be called dictomers. All right, dictosomes. Okay, or just Golgi bodies. All right, so these guys will be there now to collect, modify, and transport, like I said. So I'm just repeating on the diagram. I said they collect, so they occur in what's this, in granules or loads or just filaments. So they will be like filament kind. All right, so they can occur like in form of filaments or like this. All right, loads. All right, or just as a structure organelle like that. So they can be in these forms. All right, so filament, load, all right, or organelle. Okay, these are the way we'll be presenting, we'll be finding them. So don't get so they can be found in this form. Okay, so those are the ways you can find them. So what is their function now? What is the function? We said is to modify. Okay, one of them is just to modify, transport, and collect materials within the cells. All right, so that's about the Golgi apparatus. Okay, so they will be there to synthesize. So the word is the word here, the key word is what, to collect, modify, and what, transport what, substances that are synthesized by LAF, LAF endoplasmic reticulum, by specifically by ribosomes, when they are worked by what, by ribosomes. Okay, so ribosomes, once ribosomes work on that, then these guys will be there to what, to collect and modify, even transporting the same thing from the LAF endoplasmic reticulum, specifically synthesized by ribosomes. Okay, so the other part that we can find in this in these eukaryotes are what we call now it's what we call now or lysosome. Okay, lysosomes. All right, so what are these lysosomes? Wow, these lysosomes, they will be like a bag, in structure. Okay, so since they are like a bag in structure, so they don't have double membrane with them. Okay, they don't have double membrane. They have single membrane. There is no like we find another membrane inside. No, they just have single membrane. In short, they are small. Okay, these guys, they are more like bags. All right, that's their character. Okay, so they contain digestive and digestive tissues or just enzymes, or just dissolving enzymes. Anything that comes here will be digested. Okay, these guys, they are called and they are called what can I call them? I can call them as what? I can call them as the as suicide bugs. So they destroy themselves when they are infected. Okay, when they are infected, they will destroy themselves. All right. So uh, once they are infected, they will destroy themselves, but their function is just to do what? To digest or dissolve some what? Some things or some foreign materials. So they have that enzyme which digest or dissolve those toxic substance, like we said. Okay, so these guys, these lysosomes will be there to what? To digest foreign substance or materials when they enter. Yeah, so they can kill themselves, and also they can what? They can destroy some foreign substances. So they are good guys, but what they do is not good. Why? How do you commit yourself? I mean, how do you commit suicide? In fact, it's homicide, because they will go with every part inside them. All right, they will kill everything. So that is it. We can also talk about mitochondria now, of which it is a well-known guy in terms of what it does. Okay, so this mitochondria, it is rod shaped like that. All right. Okay, so it is rod shaped like that. So it will be found in the plasma, cytoplasm, I mean. So found in the cytoplasm. So I don't need to write, in fact, like even this. Okay, so but itself, it has what? It has double membrane. Remember, the previous guy had single membrane, but for it's one of way of what? Giving answers when you are asked to describe mitochondria. So you give to say it has what? Double membrane. Okay, okay, since it has double membrane, it contains RNA and its ribosomes. So they are it contains ribosomes. All right, so these ribosomes, we saw them in the lab. So but it is also found in what? In mitochondria. Anyway, guys, that's mitochondria. So then mitochondria. Okay, so then the other thing is um these guys, they have a membrane, right? They have inner membrane and also outside membrane. Okay, so I won't talk about it, but I will just tell you to say they have two membranes, smooth and out. Like I said, they have double membrane. All right, double membrane. Okay, so they are not like ribosomes. Ribosomes, they have single membrane. So these guys, for outer membrane, it will be there to attach what? Proteins, and also the other outside part is more like projection, so they'll be having like filaments like that. Anyway, let me not talk about that. Let's just get to know to say they have what? Double membrane, and also they have what? Ribosomes and their own DNA. Okay, so they are found in the cytoplasm as well. So that's about mitochondria. So the function will be there to produce and provide what? Produce and provide what? Provide energy required for the cell. In short, they carries what we call ATP. So production of what? Production of what? Production of ATP. So ATP is the energy readily available for cells to use. All right, so that's about these guys. They are very important guys. That's the other way is called powerhouse. That's the other name. So that's where power is produced, so it's also called powerhouse. Okay, so that's about mitochondria. So it's a very important guy. Okay, so when you are asked, make sure that you at least you describe or give these points that I've given. It's also called powerhouse. It has double membrane. It is there to what? To produce energy, produce, and also some kind of storing energy, everything like that. You explain. So that's about it. Then there's what we call now at the center. All right, at the center of the cells, there are what things that we found. Okay, so things that we found, we can find things like centrosomes. So inside centrosomes. All right, so these centrosomes, what are the functions? Okay, So they contain central organelles. Central organelles will call them things that are found at the mid. So and these things will be used in what? Cell division. So let's just say they contain two central organelles. All right. So they contain two central organelles. All right. So they have two central granules. Okay, granules. So these organelles are called centrols. All right. So they are called centrols. All right. So since they are called centrols, um, so these guys, since they contain two central granules which are called centrols, so these centrols, they are actually short. They are short. Okay, short like this. Okay, then since they are short, they have filament like that. All right, if you can view them, so they have filament. So these filaments, they will be there arranged at the right angle to each other. So if they are two like that, they have filaments that are just arranged at right angled triangle to each other, or at right angled to each other. All right, so that's about them. So during cell division, they have to move in the opposite side. So this one goes this side and taking the chromosomes. So they will be there in charge of chromosomes. So the best example that I can give you to view them, you can view them as follow. All right, so these are now centrols. All right, so they are right angled. They are two in number, like I explained. So they will be like that. So these are the filaments. So they'll be there to pull out filament once division is taking place. Okay, these are what we call now centrioles. All right. Okay, so these are what we call centrioles. Okay, so these are the filaments that will be used once we bisect to produce another cell that side. So that's how they will be going. All right. Okay, so eukaryotes, guys, are very bulk, guys, so we just have to understand, master things that I'm telling you. It's about mastering. Okay, so the other guy that we can give is the structure is what we call plasticide. All right, so this plasticide, I know you have never heard this one, but it's there in the eukaryotes. Okay, it's more like a disc like that. Okay, it's more like a disc. Okay, so it will be found in some protists. That's why it's called plasticide. So it will be found, all right, in protists, and also in some plant cells. So protist is an organism which can undergo which which is a unit which is made up of a single cell. So plant cells. All right, so it can be found in those two. So but it will be absent in animal cell, this plasticide. So plasticide, will see them, they are arranged like that. Their bag also, they are arranged like that. In fact, they will be used for photosynthesis, some kind of those. Okay, we'll find them, I'm sure, in the chloroplast, these plasticides. Okay, so they will be absent in the so absent in animals. All right, so the other thing, they'll be also absent in fungi. Okay, and also in some bacteria, they are not there. Okay, so these guys are not found. They are only found in protists, and also in some protists, not all the protists, and also in some plants. So in plants, specifically at the chloroplast, you find them as plasticide. All right, so we'll talk about them. Okay, so that's about, they contain double membrane, and they have their own DNA. So master this, they contain their own DNA. Okay, so we'll reach this at the symbiosis or endosymbiosis. All right, so they contain they contain their DNA and what ribosomes, meaning they are autonomous. It's more like they are cells on their own. All right, so they contain ribosomes. They can be found in protists and also in plants, absent in animals, fungi, and the bacteria. All right, okay, so that's about these guys. Then the other thing that we can, a good example of this plastocyte is what I said is now the chloroplast, of which now I need to talk about the chloroplast. All right, okay. So the chloroplast, what's the function of the chloroplast? They are small autonomous. So there's mitochondria and chloroplast. These guys, including now the plasticide, because they are now three. Okay, they are same autonomous. So they are same. It means they are not really fully what are independent. So these guys, they are same independence, or that's the meaning of autonomous. All right. So they are same autonomous, meaning they are cells on their own. So cells, all right, on cells on their what own. Okay, so they are cells on their own, and they don't need what, they don't need other things. Okay, that's about chloroplast. So we'll talk about endosymbiosis. I'll explain why they are autonomous like this. Okay, so we're about to finish the lesson, so make sure that you take note of these things. I'm very fast today because of the time. Okay, so there's what we call, now I've talked about a lot of organelles. So the combination of organelles, they will make what we call now the complete body of a cell or a eukaryote cell. As they make a complete body of a cell, then we need to find something like a skeleton with them. We have different type of skeletons in each living organism. We can talk of hydrochloric skeleton, hydrochloric, exo, endo, skeleton, everything like that. So now this is for within the cell, we'll call it cytoplasm. Now in the cytoplasm, the skeleton that we'll find is what? Cytoskeleton. All right, skeleton. All right, so it's also another feature. It's a combination or just a group or an association of some guys. So what are those things found in the cytoskeleton? So cytoskeleton is the skeleton within the cytoplasm. So what is the function? Okay, but before I even give the function, what are the things in charge of that cytoplasm? So they have what we call now filament. So one filament is what we call actin filament. All right, is actin what? Filament, like that. The other component that we can find is what? Microtubules. All right, so these ones will be used even they are more like centrioles as well, so the function relates to that. So all right, so microtubule. All right, so microtubules there. Then we can have also intermediate filaments. All right, so there's what we call intermediate filament. Okay, so these are what things that makes up what? Cytoskeleton. When these guys are there, then the skeleton will be maintained, making the shape and also the structure of the what? Of the cell. So the functions now that they will undergo these things, it's more like they are wires like this, okay, that support this guy mechanically in the cytoplasm. So the function is to maintain the cell shape. All right, okay, they maintain cell shape. All right, this is shape. Then the other function that it will also aid in cell division, like I said. So I'll say aid in cell division. So aid in what? In cell division, like that. Okay, then the other thing that we can also call is also support. Okay, support, transport, and modify, something like that. So they will be there to transport as they will be moving with their filaments or just loop. So they will be there to transport things within them, and also just to modify some materials. Okay, so that's about cytoskeleton. Is consisting of what? Microtubules, intermediate filament, actin filament, and the function is to maintain the shape, also aid in cell division. We saw how a centrose looks like, and those are the filament that will be used to pull out what the chromosomes, or to bisect chromosomes. Okay, so we have been talking about these guys, about autonomous things. I'm sure I talked about autonomous things. So what is autonomous, by the way? I mean semi-autonomous. There's what we call now a concept, but by the way, don't forget, when someone asks you to say what is exocytosis, okay, exocytosis, okay, when someone asks you this, endocytosis, exocytosis. So exocytosis is just substance, when substance moves out of the cell. Then endocytosis is a situation whereby things moves, things enters the cell. Okay, then here the things leaves what, the cell. Okay, that's about these terms. So make sure that you endocytosis and also exocytosis. In case you are asked, we are here to know everything. We don't want to leave any exam question, so I don't want to be blamed that you left some important points. So this is from there. Now we'll go to what we call the same thing that we are actually, it's under debate. The thing is called what, is called wait, before I even talk about that, the thing is called the term is called endosymbiosis. All right, endo simply means inside. All right, symbiosis is just relationship, or just okay. So endosymbiosis, what is this? So this is a situation whereby scientists have been discovering, finding the following organelles such as plasticide, although not really plasticide, but the two things, but plasticide is one of them, like we saw the features. So the two guys that we were under debate, or that are under debate, is what we call mitochondria, okay, chloroplast and also mitochondria. All right. So these two guys, mitochondria, we saw that they have double what? The double membrane. So meaning that they are, since they saw that they have double membrane, so double membrane, all right, it's more like they are cells on their own. The other thing that we saw it as, they have their own DNA. Okay, so the other thing that we saw, they had what? Their own what? Their own DNA and also ribosomes. All right, okay, ribosomes. So since they have their own ribosomes, then the scientists came and conclude to say, time ago, around 1297, okay, that is 5,000 years ago, these chloroplast and also mitochondria, they were prokaryotes. All right, so they said they were prokaryotes. Since they were prokaryotes, they were just, they were just swallowed. Okay, so swallowed. So this is the theorem that endosymbiosis theory. So it's the theory that I'm explaining. So it was believed that prokaryotes, I mean mitochondria and chloroplast, they came from prokaryotes. So they were just swallowed by what? By eukaryotes. So that's why we find them in the eukaryotes. What? In the eukaryote organelles or cells. All right, so by eukaryote cells, since they were swallowed by eukaryote cell, that's why they seems to be partially independent in their cells. They have their own RNA, I mean DNA, and also they have their own what? Ribosomes. All right, and ribosomes, and they have what? Double membrane. Okay, so since they have double membrane, it was believed that they are autonomous, or same what? Same autonomous, meaning same independent. They are not fully independent because they are still inside, so they just said they are same independent, although it's still under discovery. But these are the things that we should know under endosymbiosis. So this is what you should explain. They have their own ribosomes, and they can go their own cell division, such as binary binary fission. Alright, binary fission is the one which is actually relating them to what? Prokaryotes, because prokaryotes undergo what? Binary fission. Alright, so since prokaryotes go binary fission, and it was said that these guys, chloroplast and the mitochondria, came from what? Prokaryotes. So that's what they said. So thank you very much for actually attending the lecture. So we'll meet in the group. Let's meet in the group. Let's meet in the group, guys. Let's meet in the group. All right, where I'll send some question if possible. Okay, thank you for your time. See you in the next session.